Hello, good afternoon, everyone. Let's get started. We are going to continue our discussion on the um, replication of DNA. So just to quickly recap, last class we talked about different experiments that help kind of pave the way on understanding what hereditary is and the structure of DNA. So we had the Hershey and Chase experiment that showed that it was the DNA that's responsible for passing on hereditary information to the next generation. We also talked about how Ross and Franklin and Watson and Craig figure out the double helix structure of DNA molecule. And then there was also Messelson and Stahl, which did the experiment on E. coli to figure out the correct model for DNA replication, which is the semi-conservative model. So in the semi-conservative model, you pull apart the two strands of DNA, and you're going to use each strand as a template to build the new strand. So what we're going to do today in the first 45 minutes or hour is to look at the proteins and the enzymes that are involved in the actual process of replicating DNA. So that's going to be a lot of drawing here. I would suggest you do it sideways. Okay, turn your paper sideways. We're gonna start by writing something here, and then we're gonna enlarge it, and then it's gonna take up the page like that. Okay? So here we go. Let's say we have, two strands of DNA here. Two strands of DNA. And to keep it simple, I am just gonna not twist it up, okay? Just gonna keep it as an untwist model. And you might remember the DNA, the two strands of DNA, they are both complementary and anti-parallel. Okay, so DNA strands are complementary and anti-parallel. Complementary means if you know one side, you can predict the other side. A pairs with T and C pairs with G. And anti-parallel means they run in the parallel to each other. The distance between the two strands are always the same, but then the direction of the strands are opposite to each other. So one strand, we say it's from 5' to 3', and then the other strand is from 3' to 5'. It's like saying, one way of the highway is going northbound, the other is going southbound. The traffic is opposite, but the roads are parallel to each other. So nothing's new. We know this already. And what happens is when you try to copy the DNA, what you need to do is you actually need to unzip the DNA. You need to open up the DNA. Right? So like this, you're going to create a bubble. Right? So what is the bond that's holding the two strands together? Does anybody remember? What bond holds the two strands together? Like, what are these lines? What do they represent? Does anybody remember? No, not yet. That's okay. Oh, yeah, sure. Go ahead. I heard someone say something. No, maybe just my imagination. Okay, so these are hydrogen bonds, right? Hydrogen bonds hold the strands together. Okay, there are two bonds between A and T. There are three bonds between G and C, right? So in order to copy it, you have to, break apart those bonds, and you need to pull the strands apart. And over here, we create what's called a replication bubble. Replication bubble. Okay. So like, you would have the A, T, T, C, G. I'm just making these up over here. And on the other side, it will be complementary. Okay, but the point is you're going to create a bubble. You're going to open it up. So one side of the bubble is called a replication fork. Replication fork. And that is what we are going to be zooming in, okay, for our note. And that's the one that you're going to be drawing really big, okay? So it's going to look like this.
Vincent, are those letters random letters?
Yeah, I just make them up. But then the other side has to be matching, right? Like you see the A and the T pairs up, and then the C and the G pairs up, right? You see here?
Gotcha. Thank you.
Yeah. So I'm just gonna make this up a little bit bigger. Okay. So this next part here should take up almost your whole paper, okay? Like I said, if this is your paper, you got the little thing scribble on the side, and then now you have to have this big thing because we're gonna put a lot of stuff on here. Okay, so actually I don't know if this is gonna make it a little bit more complicated, but remember this is like DNA, right? So like it's kind of twisted up, right? Yeah, let me see if I can do that for you. Like, it's all twisted up, right? You understand that? Okay. Now, try to follow my arrow here first. You don't have to draw this, but I just want you to take a look here. Okay, don't do this next part. Just watch first. Okay, so look, I'm highlighting the top strands, and then if I'm twisting it, right, like that's the same strand, right? So I'm gonna say this is 5 prime, and then if I follow the yellow, it's gonna go all the way to here, and that's gonna be the 3 prime. So you guys understand that?
Vincent, where are these 5 prime and 3 prime come from?
Oh, yeah, that's a good question. I don't want to make it too complicated, but remember the what is this thing that I'm drawing? This is the phospholipid, right? Sorry, no. This is the nucleotide, right? That it has a phosphate, a sugar, and then a nitrogen base. So the carbon that is on the ring are actually numbered. Okay, so like it's like 1, 2. Actually, there might be a oxygen there somewhere, but 1, 2, 3, 4, 5. So the phosphate is attached onto carbon number 5 at one end. That's why we call it 5 prime. Okay, and the 3 prime has like yeah. So it's just the carbon numbers. Don't get too hung up on that. But you guys understand? Like I know the twist is a little bit complicated, but I'm not just doing it for artistic reason. Okay, there is a reason I do the twist here. I'll explain later. But if you follow the line when you draw your picture, if you just trace your finger right with the 5 prime here, you should see that the other end is going to be 3 prime. Okay, so like that means this is 3 prime, and then if you trace it again, just with your finger here, follow the line, follow the twist, it will go all the way to the other side, and that's gonna be the 5 prime, right? So I'm gonna just pause there and wait for you to catch up, and if you have questions, let me know. Okay, so this is gonna be bonded together, and I'm just gonna put a few letters here to help us understand as well. Okay, I'm just gonna randomly draw other letters. Okay, like it doesn't necessarily match there. It doesn't matter. Okay, maybe I'll match it. Whatever. C C T T. I'm just gonna add some more. But the key thing is they have to be complementary on the other side, right? So don't make up your own, even though you can, but just try to have what I have here. Okay. Let me know if that's okay.
I have a question, Vincent.
Yeah.
When helicase, why it's like open zip? Why it's
Yep. So we are not there yet, right? Okay. You probably have seen the notes ahead. Yep. So we're gonna get to it in a second. Okay.
okay.
Mhm. Okay. So to create the bubble to open, and this is only one side of the bubble, right? Like there's another fork on the other side, which we're not gonna look at. But we have to unzip the DNA, right? So to unzip the DNA, we're gonna need to have an enzyme to do that. Okay, and that the enzyme that's responsible for unzipping is gonna be this thing called the helicase. Okay, and that's what you were asking, right, Merritt? Helicase, C-A-S-E. So the function of helicase is to unwinds the DNA. It's like the thing that you hold on to when you want to unzip the zipper. Grab onto it, you pull down, the zipper opens up, right? So this is like that. Right? The helicase unzips the DNA, it breaks the hydrogen bond, and it creates the bubble. Right? So the helicase will continue to move this way, and then it will unzip more of it. Same thing on the other side of the fork. You're gonna have another helicase that's gonna be unzipping in the other direction. Okay. You
So
gotta.
is there any chemical reaction involved in here?
These are enzymes. So yeah, they it's I know it's weird to think of them as chemical reaction because in chemical reaction it's like you're producing new things, right? Okay. So yes and no. Okay, you're not really creating new things,
Okay.
but you're creating like a new stay here. Okay, like it was together, now you separate it, right? So unwinds it a little bit. And the process actually requires ATP, so that's why it's an enzyme. Okay, now because DNA is double helix, right? It's twisted up together. So as you unzip the DNA here, the remaining DNA is gonna have a bit of tension that's built up in it. Okay, like if it's all wound up like that, and then you're unzipping it, you're actually increasing the twist in the DNA. And if you don't do something about it, there's going to be too much tension, and the DNA might break. So there's another enzyme here. There's another enzyme that are later on, right, in the twisted portion of the DNA, and this guy is called the gyrase. Gyrase, and the job and the function of the gyrase is to relieve tension in the DNA as it is being unzipped. Okay. So like you don't want a DNA to, be all tangled up and then breaks. So the gyrase is gonna relieve the tension. You don't really have to know how it does that, but it breaks some of the covalent bond that's holding the nucleotides together, and that relieves the tension. Okay, just stop me if you have questions. Now, the two strands of DNA here are actually attracted to each other. Right? The C wants to connect with the G again, and then the T wants to go back to the A. So there is an attraction between the two strands that would cause them to come back together. But we don't want them to come back together. We want to keep them apart so we can build a new strand, right? So to separate these strands, we need more protein. Yeah. So there are these like, orange circles. Obviously, they don't actually look like that, right? But I'm just trying to draw you a diagram to help you understand. So these are what we call single strand binding proteins. And the function is to keep the two strands separated. Okay, remember they want to come back together. They have an attraction between the nitrogen base. But we got to keep them apart. We want to separate them. Okay? So those are the easy ones. We have the three things here. That's the easy one. Any questions? I know I keep on asking you, but seriously, anything before I tell you the complicated part? No, no questions? Everybody happy? All right. So let's move on a little bit. We'll come back to the diagram in a second. I just need to give you a short note. Okay. So remember, this is what happens in prokaryotes, right? Like in E. coli. Right? So most of the understanding of how DNA replicates came from the study of E. coli, right? In humans, it's similar, but with some key distinction, which I will tell you later. Okay. So the enzyme responsible for building the new DNA strand is called I'm just trying to choose a color here. It's called DNA polymerase 3. Right? ASE means it's an enzyme, so this is an enzyme that makes DNA polymer, right? And there are different versions of it. And the one that we need here is the number 3. Right? So we need to have DNA polymerase 3 to build a new strand. There are two limitations. With DNA polymerase 3. Yeah. Limitation number 1. it cannot start building new DNA on its own. It kind of not know where to start. Okay? It doesn't know where to begin. Tell you how to fix that problem in a bit. Second problem, can only build DNA in the 5' to 3' direction. On the new strand. Okay, so it can only go in one direction. So to help you understand the situation here, right? Like the first situation, cannot start building new DNA on its own. It's like my kids, right? Daily struggle. When they come home from school after they shower and all that, you know, they gotta do their homework. But you know, they don't do it on their own. They don't know what to do. Right? So I have to sit down with them. I have to do a few questions with them, and then they're like, "Oh, okay," and then they will go continue on. Nice. But I have to do a little bit for them first, with them first, right? And then I would tell them, okay, do this. Okay, then you continue. So that's kind of like this, the DNA polymerase 3. There, it's like my kids. They don't know where to start. So then what happens is you have another enzyme that's going to come along and just do a few for it. Right? So it's like, all right, I see a G here. So what does, oh, I just gave you the answer. Sorry, I was gonna ask, there's a C here. What goes with a C? G, right? It's gonna put a G there. And then there's a T here. What does T go with? Right. A, right? And then there's an A here. And what does A goes with? A goes with T, right? Okay. But here, the thing that's building this thing, it's actually building RNA instead. And I'll explain later. Okay, so instead of T, it's gonna put a U and then put an A here. So you know, it's gonna do a few nucleotides for it. This thing is called a primer. A primer, an RNA primer. That's the equivalent of me doing a few questions with my kids so that they could take over, right? They could become autonomy and do it on their own. So there is going to be an enzyme that does that. Okay, so we'll say our RNA primer is created by, there's an enzyme for that. There's an enzyme for everything. By RNA primase. And the purpose is to help DNA polymerase 3 to know where to start, okay, to continue on with the process. Right? So this helps position DNA polymerase 3. Now remember, this is nucleotide, so it also has direction. And because this is 3', this one is going to be 5', right? It's always anti-parallel. Okay, and this is going to be 3'. Okay, so then here is the polymerase 3. Okay. This is the polymerase 3. This little big triangle thing here. DNA polymerase 3. Okay. So DNA polymerase 3 sees the primer here, right? It's able to continue to build on. right? So it sees the T. It's gonna be like, oh, I know how to do this now. I just follow the base pairing rule. Okay. And so on and so forth. Okay. Except this is DNA, so like they're gonna be using T instead of A, instead of U. Okay. So it's gonna be keep on going. And remember, the second limitation here is that it can only build in 5' to 3' direction, right? So that's why we are going this way. We're going into the fork. We're not going this way because that would be in the opposite direction, right? That would be in the 3' to 5' direction. So you're gonna be copying the top strand like that. Questions?
Excuse me, Professor. I did not understand why this strand is different. For example, 4 is RNA, AUAG, and then you, Professor, use the green color for
Okay, so
another.
like I am using the different color here to show that the red part here, that is done
Yeah,
by the RNA primase, right?
RNA.
Yeah,
Yes.
and so that's like me doing a few questions for my kids, and then the RNA polymerase 3 will continue to do the rest of it. Okay, so that's why it's like different color, right? To show which one was done by the primer and which one was actually done by the polymerase 3.
Why, Professor?
Why are they
This
different?
strand is not the same, it's different.
Why are the strand different?
Yes.
Okay, because like I said, the enzyme, it doesn't know where to start. It doesn't know where to begin with the replication process. So the primer is gonna help position it. It's like, okay, you start copying here, then it goes. Without the primer, the polymerase 3 doesn't know where to start copying. That's the problem.
Okay, but AG are in just 4. It will not be 5 or 6, 7.
Oh, okay.
Or
So this is just an example, right? Like in reality, this is probably like, couple like it's it could be 20 to 50 nucleotide long, right? Okay, but like I'm not gonna write out the whole thing, right? So that's why I only
Okay.
did 4 here.
Okay, thank you, Professor.
Yeah.
Okay, got it. Thank you.
Any other question? I know this is very confusing, but like, if you don't let me know what you're confused about, I don't know, right? So yeah, ask more questions. Anyone?
Vincent, how is it that it's U and not T for the RNA primer?
That's right. So because it says it's RNA primer, right? It's RNA. There's no T in RNA, right? There's no thymine in RNA. Instead of thymine, it has uracil, right? So that's why it's replacing it with a, where there should have been a T, they are using a U instead because we're doing RNA. Okay.
Thank you.
Yeah. So some of you might be thinking, well, then wouldn't that end up having U in the DNA at the end, right? So just keep watching. Hopefully it will make sense in the end. Now, what about at the bottom? What about at the bottom? Same story, right? Same story. There has to be some kind of primer that needs to be put in place. So like, UCCG. Okay, so that's another primer. RNA primer. Okay, and look, this is 5, which means this would be 3. And this is 5. So when the polymerase come, is it going to continue to the right or is it going to continue to the left? What do you think?
Right.
To the right. So that's right. It's going to go to the right because it can only go from 5' towards the 3', right? Okay, so polymerase 3 is going to come. This is another polymerase. And then it's going to be like, A, C, and it's going to keep on going and going and going. All right. Now, what about this stuff? What about that? How are we going to copy that? Well, it turns out as you unzip more with the helicase, when you expose more DNA that needs to be copied, you're going to need to have another primer. Okay, so you're gonna put another primer here. Okay. And then the polymerase will be able to continue. Okay. And when you unzip more, you're gonna have to do more primer and more primer. It's not a problem for the top strand. The top strand can just keep on going and going and going, right? As you unzip more and more. But for the bottom strand, because polymerase 3 cannot go backwards, you need to do another primer and so on and so forth. Okay. So copy what you need first. I will show you another picture to help you understand this a little bit more.
Vincent, the helicase, it only goes in one direction or it can go back and forth?
So like there's one helicase that unzip this way, and then on the other side of the fork, there's another helicase that unzips the other way. Okay, so it's not gonna go back
Gotcha,
and
thank
forth.
you.
It's just, yeah, it's just like unzipping a zip jacket, right? Like you go down and then come back up, then you end up like rezipping it, right? Like we don't want to rezip. It's gonna unzip the DNA. Okay, so what does that actually look like on a bigger picture, right? Now let me show you in another diagram. So let's take a look at the whole bubble, and this time you can just watch. Or if you draw it, it doesn't have to be too big, okay? There we go. That's the whole bubble. We were focusing on this side before. So let's just put the numbers back in without the twists and the turns. Okay. Now I'm not gonna put the letters, but watch. Basically what happens is this. You need to put a primer here. A primer. Okay, primer. And that's like 5' to 3' here. And then the polymerase, DNA polymerase 3 is going to be able to keep on replicating. And as you open more of the DNA, you're going to keep going, right? But on the other side, if you put the primer here, that's 5 to 3. It's always opposite to the template, right? Always opposite to the black line. And this time is going to go this way, right? It's going to go this way. But as you open up more, you're going to need another primer. You're going to need another primer, and so on and so forth. Right? Same thing for the top strand, but on the other side of the bubble. You're going to need another primer and another primer. Like that, right? So basically, if you take a look at this situation here, this part is being created continuously with no breaks. Right? We call that the leading strand. The leading strand. It's being synthesized continuously, and it's moving into the fork, as in like the replication fork. Okay, so that's talking about this strand right here. The bottom one over here is called the lagging strand. And that is the one that is being synthesized discontinuously. And it's moving away from the fork. Okay, but that's only one side of the bubble. If you look at the other side, then you see the situation has switched. Right? This top strand, now top part here becomes lagging. And the bottom becomes leading.
I have a question, Vincent.
Mhm.
So, as I understood, so each DNA has the beginning and end of the strands. Okay.
Mhm.
So why this separation doesn't start from beginning or the end of DNA? Because what I can see is in the middle, the starting.
Yes, yeah, that is a good question. Okay, actually, it doesn't matter. Right? For bacteria, it doesn't matter because the DNA in bacteria forms a circle. Okay, so it's like this. This is one strand of DNA, and this is another strand of DNA, and then it's a circle. It's a circle. Right? So what that means is, like, you can create the bubble here. You can create the bubble here or here or here. It doesn't matter. In the end, it's just gonna complete the full loop, right? Right. So that's an excellent question.
So this is not for human, this kind
This
of replication.
it turns out humans are very similar, but with some differences, and I'll tell you what the differences are. Okay.
Okay.
Yeah, but as far like to Fatima's question is like, why start in the middle? Like, why not do it here, right? Well, again, I think I told you this before in lecture 3 when we talk about prokaryotes. The DNA in prokaryotes actually forms a loop like this. So this is the whole DNA. Don't draw it, okay? But this is the whole DNA. So afterwards, you're basically going to complete the whole loop, and now you end up having 2 circles, right? Identical to each other. Okay? There is another reason why it happens in the middle. And that will become more obvious when we talk about the human version. Okay. Leading strand, lagging strand. Are we good with that? Any other questions? All right. So these fragments, these DNA fragments, it's not being built continuously, right? There are little bits and pieces of it, right? Those fragments are called Okazaki fragments. Okazaki, it's the name of the Japanese scientist who discovered them, okay? Okazaki fragments. Okay, I'm gonna go back up here and I am gonna copy this section here.
I have a question, Professor.
Yes, go ahead.
Okazaki fragment, the space or the line, this, can you?
What are the
Yes.
Okazaki fragments you're asking me?
Yes.
Okay.
Is that the red one or the green
No,
one?
it's you see this whole thing is like not connected to the next part.
Yes, yes.
Yeah, those are the fragments.
Okay, okay.
Yeah. Okay, so what I did was I copy the thing from above, okay, and then I bring it down here. Okay. So what I'm trying to show you is like I'm trying to zoom in over here for you. Okay, I'm trying to zoom in here for you. Like the story is not over yet, unfortunately. Okay. So I'll give you a moment to copy that. Oh, sorry. There we go. So like, you would have something coming up from there, right? That would be like the DNA polymerase 3. I'm just waiting for you to copy, okay? I'll explain. So a couple of things that are gonna happen here. As DNA polymerase 3 approaches the RNA primer. So the DNA polymerase 3 is copying, it's making new DNA, eventually it's gonna get to the primer, right? The primer is the red thing that was laid down by the primates. Right? As DNA polymerase 3 approaches the RNA primer, DNA polymerase 3 is replaced by DNA polymerase 1. Okay, it's going to be replaced by DNA polymerase 1. So basically, DNA polymerase 1 will hop on, and it will take away the primer and replace it with DNA nucleotide. Now, just listen to me for a second. It looks like it is not necessary, right? Like, what do you mean? CGC, I take it away and then I replace it with CGC, right? So in this case, it doesn't matter. It's the same. But over here, right, the DNA polymerase 1 will take off the U, and what's the appropriate nucleotide to put on? Tell me.
T.
That's right. So it'll be like T and then CC. Okay, so replaced by DNA polymerase 1. And the function of the DNA polymerase 1 is to replace RNA primer with DNA. That's why there's no uracil. There are no uracil in the DNA. It's just thymine. Okay. Last but not least, okay. Totally running out of color here. I guess it doesn't really matter. Let's choose something else. Okay. So in the end, we have something called ligase. Ligase. Ligase is ligate means to connect them, right? So ligase is an enzyme that glue things together. The ligase is going to connect the Okazaki fragments. Right? And then the process is done. It's completed. Right. That took 45 minutes. Right? So you end up having 2 strands that are identical to the original. It is semi-conservative. You will have the original, which is shown here in black, and then you have the new one that's being built, shown in green. That's it.
Professor,
Yeah.
can you go back to the note?
Mm-hmm.
So in this process, all the RNA disappear and replaced by DNA?
That's right.
All of them?
All of them, yeah. Even though some of it's like the same, but they just have to do. Okay.
Thank you.
Vincent, what happens to RNA primer after like replaced by?
Yeah, it just falls off. They just become free nucleotides that are floating around in case you need them for another round of replication, right? Does that make sense? Okay. I mean, I know it's complicated, but hopefully by drawing it out, it makes a bit of sense. I mean, I could have just show you this diagram and talk about it, right? Which I think would be much more confusing for many of you. Okay, so that's why we try to draw things out. But you can see the key things here, right? Like what do you think number 2 is? Okay, let's label this together. Tell me what number 2 is, please. I'd be heartbroken if no one can tell me. What is number 2?
Helicase.
Thank you. I was starting to get worried. That's good. This is the helicase. And then what about number 1? What is number 1 doing there?
Is gyrase or
Yeah, gyrase. Yeah,
gyrase.
So that this guy is gonna be like removing the tangle, right? Relieving the tension, and then the helicase is gonna unzip, right? So here the square is the primase, and then the red thing here is the RNA primer. Right? And then you have the Okazaki fragments here. And you see, as DNA polymerase 3 moves towards the primer, number 3 is gonna fall off. Okay? They didn't show it here, but you know that polymerase 1 will take on the task. And it basically, in this case, is gonna remove these primer and change it back to DNA, right? They change it back to DNA. But still, there's gonna be a gap here between the Okazaki fragment, which is why you need the DNA ligase to glue them back together. Okay? No Okazaki fragments on the top. It's able to be synthesized continuously. What do we call that strand again if there are no breaks? What do we call this top strand? That there are no breaks in it.
Leading
Yeah.
strand.
That's right. That's a leading strand. Right? It's going faster, right? Like there's no break. And then the other one's kind of, you know, you got to do it bit by bit, which is a little bit slower. So that's why it's called the lagging strand. Lagging strand. All right. That's it. Now, why do we have to know this? Well, actually, if you had a PCR test done on you during COVID, the PCR technology is possible because of the understanding of how DNA replicates. Okay, so we're going to talk more about PCR tests later on in the course. But it's, again, based on the understanding of this. You are basically trying to do the same thing in a test tube when you try to do PCR. Okay? So that's that. Any last minute question before we move on?
We need time to digest it.
I know it's a lot of information. I'm so sorry. I feel very bad. I do. But unfortunately, it's you know, it's part of the curriculum. All right. So that is that stuff is what happens in E. coli, in bacteria, which are prokaryotes, right? So for humans, it turns out it's very similar. Right? The process of DNA replication is similar in eukaryotes, but there are a few key differences. Okay? For one, eukaryotes, including humans, we don't have circular DNA. Okay? You guys understand what I mean when I say circular DNA? It's like their DNA is double helix, okay? But then it goes around. I don't even know how to draw this properly. Okay, but it goes around to form a circle, right? Like this. Oh, okay, you don't have to draw it, but it's like the DNA, but then it loops back on itself. Okay, so we are not like that. Our DNA are linear, right? Linear. It doesn't loop back on itself. Okay, so it's really long, and if we start at one end and then just unzip it all the way to the other side, it would take too long. Remember, we have like 3 billion base pairs that we need to copy. So what happens is in humans, okay, I'm just gonna draw it regularly again. You don't have to draw, you can just watch. In humans, you're gonna have multiple replication bubbles. Okay, you can have it, in fact, up to 100,000 replication bubble. So then you can have multiple copying sites at the same time. Imagine I ask you to copy the encyclopedia. Okay, not even encyclopedia. Let's say copy the textbook, right? Like if you start from page 1 and you do it by yourself, it's gonna take forever. Okay? But if I say the whole class, we're gonna do it together, right? Then you start in chapter 1, you start chapter 2, you start chapter 3. Everybody do a little bit, and eventually we have the complete thing. We have the whole thing copied, right? And that's kind of the point here. By having multiple replication bubbles, by having multiple origin of replication, you can speed up the process. Okay, so this allows faster copying. All right. Here's a picture of it. The purple here is the parent DNA, the original DNA. You're gonna create these origin of replication, creating the bubbles, and then each bubble will expand laterally, right? With the DNA polymerase going into the fork, away from the fork, and then the bubble is gonna get bigger and bigger, and eventually this bubble is gonna merge with that bubble. And when all the bubble merge together, you end up having two separate DNA. So this is one DNA, two strands, right? One DNA, and then this is the second DNA. Right, first DNA, second DNA. And of course, they are identical. And more importantly, is semi-conservative. You see, half of the strand is the original purple, and then the other strand is the new blue. Two identical copies. Okay. So that's the first difference between prokaryotes and eukaryotes. It's like how many origin of replication it has. Second difference. Most of the enzymes in prokaryotes have a eukaryote counterpart. What that means is, there's a helicase in prokaryotes. There will also be a helicase in human. There are ligase in prokaryotes. There will be a ligase in human as well. But sometimes the enzymes are represented by multiple enzymes. So, there's polymerase 3 and polymerase 1 in prokaryotes, but then in humans, there are 5 different ones. Okay, 5 different human DNA polymerase 3. Okay, which we're not going to learn all of them. Okay, but some proteins have multiple counterparts. So that's the second difference. The last difference is that some DNA are lost during each DNA replication in eukaryotes. Every time we copy our DNA, we lose a little bit. So it gets shorter and shorter and shorter and shorter. This is true. Right? And the question is why? I'm just debating if I should tell you why now or after the break. You know what? We had a heavy hour. Okay, let's just take a quick break. You drink some tea or something, and then when we come back, I'm gonna try to explain this to you. Okay, so we'll resume at 1:30. Right? See you in a bit. All right, and we are back. So we were talking about the differences between prokaryotes and eukaryotic DNA replication. So there are multiple replication bubbles in eukaryotes. There are more enzymes involved. And the last difference is the fact that every time we copy our DNA, it gets shorter and shorter and shorter. So let's take a look at why that happens. And to look at why that happens, we kind of have to, draw it one more time. So let's say, again, we have the linear DNA. So let's say this is our entire DNA. It doesn't loop back to form a circle. Okay, so let's say we just have the bubble here. 5 prime to 3 prime, 3 prime to 5 prime. You have this diagram already, but you know, it doesn't hurt to draw it more times to, you know, get better at it, right? So let's just focus at the bottom strand right here. Right? So we already understand if you put a primer here, then the polymerase is gonna go this way, right? So that's fine. And it's gonna go all the way to the end, basically, right? That's fine. Over here, we're gonna need another primer, and then it's gonna go here. And listen carefully. The polymerase 3 will fall off, but number 1 will come, and then it will replace the primer, right? So this red stuff is gonna become regular DNA. Same thing here. Number 3 is gonna build, and then it's gonna fall off. Number 1's gonna come and replace it. Yeah, same thing here. It's gonna build. It's gonna fall off, and number 1's gonna replace it. But there is a problem when we get to the very end. Okay? This part, or I should say this RNA primer will not be replaced because nothing is coming from upstream, right? Like that, this one's gonna get replaced because something is gonna be coming down, right? Same thing, like this one's gonna get replaced because something is coming down. But like if you are at the very end, nothing's coming. Okay? Like without the number 3 coming, the number 1 doesn't know to get on, right? So nothing is coming and it will be lost. Okay, so this part of DNA will not be copied and is lost. Okay, same thing on the other side. Right? You're gonna have the primer. And that's gonna go this way, no problem, all the way to the end. Right? But this part will be lost. So every time we copy, we chop off the end a little bit, and a little bit, and a little bit, and a little bit, and a little bit. So our DNA gets shorter and shorter and shorter. I know you must have a lot of questions about why this is not a problem, or like, is it a problem? Just keep listening, okay? I'll explain. But before I explain more, is it clear why we are losing DNA at the ends?
For me, it's not clear. I have something because maybe I will say you will correct me. Maybe because of the end, the 3 prime is on the end. That's why we are losing DNA, or why?
Okay, so I want to ask you then. You understand this red part is RNA, yes?
No, it's DNA.
No, the red part. The red part is RNA, right?
Yes.
Okay, and it's gonna become DNA, right?
Yes.
Why is that? Because there is a polymerase that's gonna be coming, and then the polymerase 1 will get on and then replace it, right?
Yes.
So that means something has to be coming down to
Okay.
trigger
Yes.
the replacement, but nothing is coming. This is the
Nothing.
end of the line.
Yeah, I got it. Thank you.
Yeah. Everybody else okay? Of why we are losing DNA? Now, this is not a problem, for circular DNA. Okay, so here's a circular DNA. Okay, like it forms a loop, right? So for simplicity, I'm just gonna draw one primer, okay? Like there are gonna be other ones as well, okay? But for simplicity, I'm just gonna draw one DNA and one primer. Watch. You're gonna build, keep on building, and eventually you're just gonna loop back on yourself, and then you can replace the primer, right? So no DNA is lost. This is not a problem for prokaryotes, but it's going to be a problem for humans because we have linear DNA as opposed to circular DNA. Okay. So is this bad?
Professor,
Yeah, sorry, go ahead.
excuse me, what kind of problem?
Yeah, I'm gonna tell
It's
you.
kind of disease for human?
Not quite, okay? So I'm gonna tell you what happens now.
Okay,
Now,
thank
obviously, it's bad to lose DNA. We don't want to be chopping off DNAs, but it does happen. So, to protect the ends of a DNA, we actually have a bunch of non-coding DNA. Okay? So coding DNA means these are DNA that are important. It gives instruction to make proteins, for example. Those are coding DNA. But at the ends, because we know we're going to be losing some genetic material every time, we have some non-coding DNA at the ends. These are called telomeres. Okay, telomeres. And they are just a bunch of repeats of TTA, GGG. So at the ends of your chromosome, it's TTA, GGG, TTA, GGG repeated like hundreds and hundreds of times. So even though we're losing a bit of DNA every time, but we're losing the non-coding stuff. We're losing information that are not important. Okay? So you can think of the telomeres as the plastic thing that is at the end of your shoelaces. Okay? Let's say every time you tie your shoelace, we cut off a little bit of the thing. Okay, so you know you cut it off a little bit. That's okay. You're not gonna affect your shoelaces. You cut it off, cut it off, cut it off. But eventually, if the whole plastic thing is gone, then the shoelace is gonna start to fray, right? And it's gonna damage the shoelace, right? And that's what happens. The telomeres, they protect the DNA from fraying, and it delays the shortening of critical information during DNA replication. So these are chromosomes that you're seeing, real chromosomes under the microscope. In this particular picture, they put something to cause the telomeres to light up. Okay, so you see at the end, right? The white dots, that's where the telomeres are. Okay? So you have it on both ends. When the telomeres become critically short, then the cell will not be allowed to divide any longer. Okay, because the cell knows if I do another round of division, which means I have to copy all my genetic material. If I do that one more time, next time when I lose the DNA at the end, I'm going to be losing actual information. Okay, the protections are all gone now. Right? The telomeres after, 30 and 40 rounds of division, we have no more protection. We cannot afford to do another round of division. So when that happens, the cells no longer divide, and we say the cell becomes senescent. Senescent. Okay? It doesn't divide anymore. It's old now. Right? So depending on the cell type, it might stop dividing after 40 times or something like that. So as we get older and older, the pool of senescent cells in the body becomes bigger. And that is in part what contributes to aging. Any questions about telomeres? Like, what are they and what are their purpose? Are we clear on that? Okay. So I want to talk a little bit about Dolly. Has anybody heard of Dolly before? Just give me like a thumbs up or like, just type a yes in the chat.
I watched the video you put on the.
Oh yeah?
Yeah.
That's good. That's good. Okay, so some of you might have heard of it before. For those of you who don't know, Dolly was the first mammal that was cloned, okay, from an adult somatic cells using a process called nuclear transfer, but it only lived 6 years, half of the expected lifespan of a regular sheep. Okay, so how does it work? I want to ask you, how many sheeps do you think it takes to clone Dolly? Okay, I'm not talking about like the fail attempts, okay? Like the one time that it worked. How many parents do we need to clone Dolly? Any guesses here? No guess?
One.
One? Yeah, okay. Yeah. More than that, okay? So let's take a look. So this is Dolly. Okay. So here we have a sheep. Okay. So this sheep here is called the nucleus donor. Okay. So we take what do we take? We take a somatic cell. A somatic cell is removed, and the nucleus is extracted.
What is somatic?
Yeah,
So-
somatic means any cells in the body except for the sperm and egg. Okay, so it's a somatic cell, a bodily cell. Okay, maybe it's a muscle cell, maybe it's a liver cell. Okay, so they take a somatic cell from the dolly, and then it removes the nucleus. Okay, nucleus. Okay. So of course the nucleus contains all DNA, right? So that's your parent number one, so to speak. And then we need to have another sheep. I'm sure they're not very happy to be in this experiment, but let's just assume they are. So number two, this is the egg donor. Egg donor. So female, right? So take the egg. This is an egg. And then this is the nucleus. So an egg is removed, and the nucleus is removed as well. Okay. So now you have just the empty egg. So egg normally contains half the genetic material, right? Because the sperm would have the other half, and when they come together, you get the full set. But in this case, you remove the nucleus, so you just have the egg, just the cytoplasm and everything in it. Okay. Then what happens is they put the nucleus from the donor into the egg. You can do this under the microscope, okay? So the egg, the nucleus from, we'll call this sheep 1 and sheep 2, okay? From 1 is inserted into the egg from 2. The result is an egg with full set of chromosome. Egg with full set of DNA. it is as if the egg got fertilized. Do you understand? Because normally the egg only have half sets, right? To get the missing half, you need the sperm to fertilize the egg, right? Then you have the full set. But in this technique, they remove the nucleus from the empty egg, and then they put the nucleus from the donor, which has the full set of chromosome already, and they shove it in the egg. And now we have an egg with all the DNA. Right, so far, 2 sheeps, right? Okay, where do you think they're gonna give this egg to? What do you think? How do they grow this? What do we call it when you need something to grow a baby on behalf of someone else?
Donor egg.
Sorry, what is that again? I didn't quite catch it.
So I think that they put this egg inside body of sheep too.
Yeah, okay. So actually they put it inside the body of sheep number
Surrogate.
3. Yeah, sheep number 3. But what do we call people? I mean, humans do this, okay? Like if a person cannot have baby, they sometimes would have someone carry their baby for them, right? Do you know what we call?
Surrogate.
That's right. So this is a surrogate sheep. Okay. So the egg is implanted into a surrogate. And then later comes the dolly. Okay, so this is dolly. So here's the question. Dolly is the clone of who? Is dolly the clone of 1, 2, or 3?
1.
That's right. It's the clone of 1. Because all the genetic material is basically contributed by the nuclear donor. Okay, so that's how they created the clone. It's genetically identical to the nucleus donor. Okay, the egg was merely there to provide a place to put the nucleus in, and the surrogate provides the womb to grow the sheep, right? So that's how they cloned the dolly. Now, dolly only lived half as long as you would expect for a sheep, right? And this is the reason. And listen carefully and see if you can follow. The sheep that donated the nucleus, it was an adult sheep. Okay, so that sheep has been living for some time. I don't know how old. Let's say 6 years already in its life. Okay, adulthood is like different for animals and humans, right? Okay, so 6 years is a long time for an animal, could be. So that means all the cells in that sheep had been dividing for this many years, right? And so the telomeres have been shortening. Right? So let's say, just theoretically speaking, at the time when they took out the nucleus, let's say the cell has been dividing for 20 times already. So when they transfer the DNA into the egg and then let it grow in the surrogate, dolly was born. So, age-wise is like age 0 because it's a newborn. But on a cellular level, the cell would have the same biological age as the original donor. Because those cells has been dividing for like 20 times already, dolly cells would be born with the same biological age. It is as if the cells have been living for 6 years already, undergoing so many cycles of division. So what happens is, because the telomeres are already shortened, dollies are born with these shortened telomeres. The cells are not allowed to divide too many times. So instead of having, let's say, 40 rounds of division, Dolly was born with only 20 more cycles to divide before the cell becomes senescent, right? Before the cells become too old to divide. And so even though it's a newborn, but biologically is already old. So that's why the lifespan is shorter, half of the expected lifespan of a sheep. Do you understand?
Yeah.
Yeah. So that actually creates a question, right? How come that doesn't happen in humans? So, for example, like an adult, let's say they have a kid when they are 30 years of age. They had their first kid. But at that point, the person has been living for 30 years. The cells have been, dividing for 30 years, have gone through X number of division. Then how come the baby is not born with shortened telomeres, right? Because after all, don't we pass on our genetic information, right? How is it different? Well, fear not, because there is an enzyme, right? This enzyme is called telomerase. Okay, ASE is the enzyme. Don't confuse telomerase with telomeres. Telomeres are the repeats. That's the plastic thing at the end of the shoelace, right? Telomerase is the enzyme. So telomerase are going to be turned on in germ cells. Okay, germ cells, it's not what you think it means, okay? It's not like bacteria. Germ cells are cells that becomes sperm or egg. In male, it becomes sperm, in female, it becomes egg. So these are the cells that would become the sex cells. And so the telomerase is turned on in these cells so that you can rebuild the telomeres. Without the telomerase, telomeres get shorter and shorter and shorter. But with the telomerase, you can maintain the telomere length. So that even though your telomeres get shorter, but when you want to have a baby, the sperm, creating the sperm and egg, thanks to the telomerase, you recreate the telomeres. It resets itself. So when you pass it on to your baby, the baby is born with brand new telomeres. And then it's going to be able to start from fresh. Okay. So what do I mean by telomerase is turned on in germ cells? So last class, right, Fatima asked, like, do all the cells have the same DNA? Yes, they do. Okay, but like in the liver cell, we don't turn on telomerase, even though you have the gene for it, but we leave it off. Okay, we don't turn it on. Right? So it's only in the germ cells that we turn on the telomerase so that we can reset the telomere length. Okay? So usually people would ask me at this point, well, I said we have the telomerase gene in all the cells, but they are not turned on. Would turning it on be beneficial then? Let's say we turn it on in our skin cell or something, then wouldn't the cells be able to divide and divide forever and not get old? Would that be able to, extend human lifespan? Well, the answer is probably not, because telomerase actually does get turned on in some cases, but by mistake. So if your body accidentally turns on telomerase in a cell that is not supposed to turn it on, that actually creates a condition that would help lead to cancer. Okay? So what is cancer? Cancer essentially is uncontrolled cell division. The cell keeps on dividing and dividing and dividing, and there's no way to stop it. Right? I told you in lecture 3, the HeLa cells, right? Those are cancer cells that are taken out from the from Henrietta Lacks, right? The person with the ovarian cancer. And you can virtually grow them forever in a petri dish. So that's the same thing in the body. The cancer cells keep on growing, and there's nothing stopping it, and it takes up all the resources from the body and kills the person, basically, right? And one of the reasons why cancer cells are allowed to keep on dividing is because they turned on the telomerase. You're not supposed to have telomerase in regular cells. You're only supposed to have it in germ cells, right? But when you turn it on, then the telomeres keeps on rebuilding and rebuilding. So you can keep on dividing and dividing, and the cell never becomes senescent. Okay? So when they look at various tumors, they found that telomerase is almost always elevated. And so researchers are exploring telomerase inhibition as a potential treatment for cancer. If we can block the telomerase in these tumor cells, then maybe we can stop them from dividing. Okay, and then combine this with like radiation therapy, maybe you can shrink the tumor and destroy it, right? So that's that. Okay, just gonna go through a few questions here with you guys, and then that would be it. What is the key difference between DNA replication in prokaryotes and eukaryotes? Okay, so let's see. Prokaryotes have telomerase and, have telomeres, and eukaryotes do not. That is false. It's the other way around. Right? It's the other way around. Right? We are the one that needs the telomeres to protect the ends of the DNA, right? Prokaryotes don't need them because they have circular DNA. Prokaryotic chromosomes have a single origin of replication, whereas eukaryotic chromosomes have many. That is true. Okay, we want to open up many bubbles so we can do it faster, right? Prokaryotes produce Okazaki fragments during DNA replication, but eukaryotes do not. We both have Okazaki fragments, right? Because of the 5' to 3' limitations, right? RNA primers are only needed in prokaryotes, also not true, right? Both prokaryotes and eukaryotes, we need the RNA primers. Okay, this one is pretty short. Maybe you guys can try it. Let me do the polls. Go ahead. Okay, the correct answer is ligase. Okay, ligase, to ligate is to glue things together, is to join things together. So ligase connects the Okazaki fragments. RNA primers are removed by, okay, so if you have been following, it is going to be the DNA polymerase 1. Remember, as number 3 approaches the primer, it falls off, and the number 1 gets on and replaces the primer with DNA, right? Last question. Telomerases, which are the enzymes, are needed to re-lengthen shortened chromosomes in some cells. That's correct. But I see all of the above, so I gotta keep reading. Excuse me. Prevent cancer development. That's not true. Okay. In fact, having telomerase in body cells, that leads to cancer, right? So, A is the answer. Remove non-coding repeats of TTA, GGG. No. The the telomeres are TTA, GGG. So if anything, telomerase actually adds the TTA, GGG, right, to rebuild the telomeres, right? They do not shorten the ends of the chromosome, they actually make it longer. Okay, so the answer is A. I think that's a good spot to stop. There are still, we only have, there are 72 slides in this PowerPoint. We've gone through 32 of them. Okay, so still have 40 more to go. But that's okay. Like it's, it's a lot of information, so it probably takes some time to digest it. Just before you go though, let me quickly take a look at what's coming up so that we have an idea of what to expect. There we go. Okay, what do we have? We are March 6th today, and we did not finish DNA replication. That's fine. We will finish it next class though, and probably start on the protein synthesis. Okay, so protein synthesis is going to take some time, like 2-3 lessons. So we're gonna have the microscope lab on March 18. But instead of like not having class, we're gonna need to have a class. Okay, so like the microscope lab, you're just gonna have to do on your own time during that week. This one is much better than the beetroot one. Okay, because a lot of you were emailing me saying you didn't have enough time for the beetroot. This one is not done in a quiz setting. Okay, so it's like a report that you fill in, so there's no time constraint. As long as you hand it in by Wednesday, you should be okay. All right, so hope that helps. Yeah, that's it for now. Okay, thank you. I'll see you next week. Have a good one, guys.
Thank you.
Thank you.
Bye.
Bye.